Oh, oh, all right, everybody. What we are going to be getting into right now is I'm going to circle back on some different information about mechanisms and enzyme mechanisms. Um, we had exposure to some exposure to a acid-base catalyzed reaction, as well as the mechanism for a serine protease. I want to bring in a couple of other mechanisms, and one because I think that it's a really great model for metal ion catalysis, and then another one because I think that um, it's another good mechanism for covalent catalysis, and that is the lysozyme mechanism. We'll dig into that quite a bit and look at some things from different angles. But until then, let me do one last thing. Perfect. Yeah, that's good. That's what I wanted. Okay, great. Okay. Sorry, I need to go full screen. Then I need to share screen and booyah. There we go. Okay, so mechanisms of enzymes. We've already kind of seen these, but we're going to go a little bit deeper into them. So um, we need to be able to identify different strategies, give examples. To me, one of the most important things about any sort of enzyme mechanism is recognizing the players that are involved, recognizing specifically the amino acids that are involved and what they're behaving as, what they're acting as. So the first type of mechanism that we're going to look at is a metal ion catalyzed reaction. Now, this is basically a model or an enzyme, which is going to have NNN, a divalent cation, and then a water molecule. Okay, that's cool. Then, if you look at the kind of end state here, what we have is a hydroxide ion. Okay, so effectively what happened was that metal ion was able to kind of attract that oxygen, that the lone pair of electrons from that oxygen within that water, and and essentially get that water to lose a proton or to lose a hydrogen. Now, this is a great model for metal ion catalysis, and this, in fact, the model for the enzyme known as carbonic anhydrase. Carbonic anhydrase is an enzyme that takes H2O, CO2, and produces HCO3 minus. Now, what we're seeing right here with this IM, well, IM is representative of an imidazole ring, and this would be a histidine residue. Histidine is an amino acid that has a five-membered ring, and the functional group of that five-membered ring is imidazole, or the functional group name for that five-membered ring is imidazole. So essentially what this does is it uses deprotonated histidine residues to basically lure in or attract the zinc ion, the Zn2 plus. What that zinc ion is going to do is it's going to bring a water molecule in. And as soon as it brings that water molecule in, what's going to be left behind is a hydroxide ion. So essentially, if you want to look at this as like the starting point, look down here. So we have water coming in, the hydroxide is attracted to Zn2 plus, and a proton is released. Okay, so just imagine that that has happened right here. Because what's to visualize this figure, basically what we've got is these steps that kind of go on and repeat over and over and over and over again. Now, with that in mind, a fantastic question is how? Why? Well, let's think about this. H2O is going to lose or is going to become OH minus and H plus. Why is that? Well, the reason for that is this zinc ion, whenever it brings that water molecule, the pKa of water, of that water molecule that's bound to that zinc 2 plus, is going to go from 14, it's going to drop all the way down to 7. Now, that's the sort of thing that you might be thinking like, I'm having trouble wrapping my mind around that. The way that I always like to think about this is water with a pKa of 14. If it is in a solution with a pH of 7, essentially 100% of water molecules with a pKa of 14 will exist as H2O. Meanwhile, if by comparison we've got water that drops, that's pKa drops to 14, or sorry, to 7, and is in an environment with a pH of 7, well then about 50% of those water molecules are going to go to OH minus. Because we know that whenever we have something where the pH is equal to the pKa, we have an equal concentration of the protonated and the deprotonated form of that substance. If we look at H2O as the protonated form, well, then we're going to have 50% of those water molecules looking like that, and 50% of them that look like this. So what effectively is happening is this zinc ion, or this kind of zinc capturing this H2O, causes the pKa to drop down to 7. Okay. Now, what that allows is that allows us to lose that proton. This hydroxide ion with a lone pair of electrons can then go to this carbon in the center of carbon dioxide, which is going to inevitably force some of these electrons within this double bond between carbon and oxygen out to this oxygen. So that oxygen is going to have a negative charge. So we're going to have a molecule that looks effectively like this, which, I mean, correct me if I'm wrong, but let's count this out. We've got a hydrogen, a carbon, an oxygen, oxygen, and oxygen. So that's 1 hydrogen, 1 carbon, 3 oxygens. Effectively, we've formed HCO3 minus. That HCO3 minus is a little bit more stable and able to act on its own. It's going to lose, or it's going to be basically released from the active site of imidazole, of the carbonic anhydrase, and water can take its place. And that whole process can begin over and over again. Now, that's metal ion catalysis. What we see with carbonic anhydrase is the pKa drops from 14 to 7. Awesome. Now, substrate binding facilitates catalysis. This is an important point because I always kind of think about this like, if you have 2 reactants and they are in close proximity together, there's a chance they will react with one another. Now, let me draw a parallel here. Let's imagine that you have 2 friends and you think that they would really get along well together, and you've decided that, maybe they should date one another. And so what you're going to do is you're going to play matchmaker, and you're going to say, okay, my friend A and my friend B, those 2 friends are going to meet up and they're going to fall in love, and it's going to be fantastic. Them falling in love is the reaction here. Okay. So if you think that, then by virtue of them knowing you, they might bump into each other at some point. You might have them over for a Super Bowl party or I don't know, sports watching party of some sort. Okay. That could happen. Now, that's like 2 reactants that if they are next to one another, they may react. And an enzyme is going to bring those 2 reactants in close proximity, and their chances of interacting are greater than if that enzyme is absent, where those 2 things are just kind of floating around in the ether. Now, let's say that you really want this friendship or this courtship to take place. What you're then going to do is you're going to say, okay, I want you to both meet at Costco. And so you're going to provide like a little bit, you're going to kind of stack the deck in the favor of them meeting. Well, an enzyme is going to do the same thing. Enzymes bind substrates in the correct orientation for a reaction. And whenever that takes place, well, the reaction, the enzyme doesn't have to move or anything. Essentially, what happens is these two reactants are facing one another. They're ready to react with one another. That reaction is going to take place, and it's more likely to take place, and it's going to happen faster than if the enzyme simply binds them and they're facing in opposite directions. When they're facing in their position in the correct orientation, their chances of reacting are infinitely higher. Now, when an enzyme freezes translational and rotational motions of both substrates and catalytic groups, your rate is going to increase up to 10 to the 7th fold. Okay, so now this is like you playing matchmaker, but you have said, okay, you're meeting at Costco. You're meeting at the food court at Costco. In addition to that, this is you like subliminally putting this information into your friends' heads. You're saying, you are ready to meet the love of your life. And you're kind of like, I don't know, you're just like imprinting that into their brains. So then those people are like, okay, well, I'm ready to meet the love of my life. They meet at Costco. They're facing one another, and then all of a sudden they're like, everything is perfect. The stars have aligned. I'm ready. These substrates are effectively the same way. You know, when they are facing one another and all of their atoms and electrons are in the correct rotation and correct positioning so that they can react with one another, the rate enhancement is 10 to the 7th fold. So we're going to see them go faster, and we're going to see them go faster than faster by this much. Now, preferential transition state binding, this to me, I had a professor as a graduate student who had this model, and he said, okay, well, here is our reactant. We're going to call it just this stick, this line right here. And this is our reactant, or our substrate, whatever, substrate, reactant, whatever you want to call it. And here's our product. And he kind of looked at it. He would bring a stick out. He'd bring a stick to class, and he'd say, okay, well, we need this stick to go from, you know, a straight line to broken in two pieces. And everyone would look at his hands and say, well, your hands are the enzyme. That's broken the stick in half. And he's like, you're right. You're absolutely right. But if you did not have that enzyme there, what are the chances that that reactant spontaneously breaks? You know, we would all just say, oh, it's very low. Okay, so then he said, all right, well, let's say that I agree, my hands are the substrate. And let's say that as the enzyme, sorry, my hands are the enzyme. If my hands, and I'm going to use a pen as my reactant or my substrate, my hands bind onto this perfectly. What's going to happen? Well, what's going to happen is, you know, my hands are going to move. That's going to break the stick. And he would say, you're right. But in order for that to happen, there has to be some force that causes my hands, the enzyme, to move. And everyone would be like, yeah. Well, that requires some sort of force, that requires some energy input. And he said, all right, well, how about as an alternative? And good news, everyone, I've got a pencil, so I'm going to sacrifice this pencil. Actually, you know what? I'm not. I'm going to use a straw. Got a straw here. And do I have scissors? Afraid I don't have scissors. But let's imagine that this is our stick and it's broken in two. There we go. He said, well, let's imagine that if, we can see our substrate go from this to bending to this snapping, okay? Would everyone agree that this position right here, let's imagine that if we put enough force, this substrate would break. This right here is a transition state because we got to go from here to broken in two. This is it's transitioning to that broken in two point. And everyone would agree with that because this right here, this positioning of our substrate is not this. It's also not broken in two. So you'd say, okay, that's kind of your middle ground. Well, what if we had an enzyme that its structure was not, oh, I'm going to grab onto this, this thing in two places, but rather it would grab on to the substrate, or it would only have an affinity for the substrate in this transition state. Like this right here is the structure of my enzyme. Well, is this structure going to bind this right here? No, it's not. However, and this is hard because I'll only have two hands, this structure of my hands does have an affinity for this kind of permutation of my substrate. So if the structure of my enzyme resembles or has a kind of curve to it or a shape to it that resembles or complements the transition state, that's going to increase the rate of my reaction because my enzyme's not going to get trapped holding on to it like this. Instead, my enzyme is only going to bind it when it's 75% of the way broken. Now, this binding is usually the major rate enhancing effect due to straining the substrate to look more like the transition state and increasing the transition state concentration. So it's basically, it's almost like saying, hey, substrate, I'm the enzyme, I will bind you, but you've got to look like the transition state. If you don't look like the transition state, I can't really help you. Now, when it comes to studying enzymes, and in particular covalent catalysis, there we are. There are groups that are important to look at. I always said nucleophiles and electrophiles, or nucleophiles and electrophiles. Your nucleophiles are typically a part of your enzyme, whereas your electrophiles are typically your substrate. Now, this list of nucleophiles has our amino acid side chains and then has them in their nucleophilic form. Our nucleophilic form, or our nucleophiles that can form this great nucleophilic form, have a hydroxyl group. Which amino acids have a hydroxyl group? Serine, threonine, tyrosine. Or they have a thiol or a sulfhydryl. Well, cysteine is the only one that really has that, because methionine is going to have a methyl group. RNH3, well, that's going to be an arginine, a lysine. And then this last group down here, this ring, this is our imidazole ring. This is histidine. So STYCRKH, those amino acids are most likely to participate in covalent catalysis. And they are most likely going to be serving as nucleophiles. Your electrophiles, by contrast, these are parts of your substrate, or these are a part of your substrate. You have a metal ion, carbonyl carbon, cationic imine. Those are going to be the players that are your electrophiles. Now, we've already talked about the serine proteases and the catalytic triad. Well, chymotrypsin is another example of a serine protease. And here what we're going to do is we're going to talk about different serine proteases. There's a number of different serine proteases. We'll talk about a couple, then we'll talk about a couple more. Now, what makes these serine proteases different from one another is their ability to recognize different substrates and hydrolyze the adjacent peptide bond. Now, this is something that people always struggle with. I struggled with it. I've had students who struggled with it. I will continue to have students who struggle with it. And I don't remember why I struggled with it. And as an instructor, I've tried different things, but hear me out. Chymotrypsin is a protease. So it is going to cut peptide bonds. Now, it is going to cut peptide bonds after a specific amino acid or after specific amino acids. When you look at this polypeptide down here, this has a total of 6 amino acids. It has a sequence of A, F, N, S, M, E. Now, there are 2 amino acids that are highlighted, phenylalanine and methionine. In addition to that, there are 2 bonds that are highlighted. They are in magenta right here and right here. If you look at those bonds, what you'll notice is that there's a carbonyl carbon going to a nitrogen. Carbonyl carbon going to a nitrogen. That is a peptide bond. That is the peptide bond that chymotrypsin will cut after. Now, if you look at the rest of this molecule and try to find another carbonyl carbon going to a nitrogen. Here's a carbonyl carbon. Here's a nitrogen. This right here is a peptide bond. Chymotrypsin is not going to cut that. So chymotrypsin is not just willy-nilly cutting every single peptide bond, it's cutting specific peptide bonds. The specific peptide bonds that it's cutting are after amino acids like phenylalanine and methionine. What it is looking for is a bulky hydrophobic residue. Phenylalanine and methionine are both bulky hydrophobic residues. So the way that I like to ask questions about this are chymotrypsin will cut after, or it will take this polypeptide. And what will it produce? Well, it's going to cut here, which is the same as right here. Then it's going to cut after methionine. So it's going to cut here and here. So the polypeptides that are going to result from chymotrypsin cutting this are AF, that's a dipeptide, NSM, and E. Those are the polypeptides that will result. Now, it's not a trivial thing to understand, but serine proteases all have very similar properties because they love to cut after that. Now, serine proteases, well, chymotrypsin is one serine protease. Trypsin is another serine protease. Elastase is another. All of these serine proteases, and all serine proteases for that matter, have this particular pocket. This is known as the specificity pocket. Now, if you look at, I always like to use trypsin as my example. Trypsin has a negatively charged amino acid. That's an aspartate in its specificity pocket. Now, what do we know about negatively charged groups? They like to attract positively charged groups. So if we look at, let's say, let's go with, okay, give me just 1 second. Let's go with, I'm going to go back to this previous peptide. RAF, oh, NSME. This peptide. We already saw that if we took, and okay, so if we take this peptide and we give it to trypsin, within the specificity pocket of trypsin, we have a negatively charged residue. This right here, RAFNSME, this is the substrate. That substrate, provided that it has a positively charged residue, that positively charged residue is going to fit right in here. Well, does this have a positively charged residue? It does. Arginine. Now, what trypsin is going to do is it's going to see this peptide and it's going to say, aha, I have a positively charged residue in my specificity pocket. And I am going to cut after that positively charged residue. So trypsin will take this peptide and make R as one peptide. And so here are my products. And the other peptide is AFNSME. Okay. Now let's go on to take the same peptide and throw it at elastase. Now, if you look at elastase and you look at the specificity pocket, what I want you to notice is that there's a pair of residues. There's a valine, residue 190 and residue 216. What do we know about valine? Valine is a hydrocarbon. Valine is a hydrophobic residue. And so valine as a hydrophobic residue likes to be around hydrophobic residues. Now, in addition to that, if we look at this specificity pocket, I would put this at almost like 40% of the space of this specificity pocket is occupied by valine. So what that means is that a smaller amino acid will fit into this space. Larger amino acids are not going to fit into that space. So if we take a polypeptide like RAFNSME, we need a small hydrophobic residue to fit into this space. Is arginine, and when we think about hydrophobic, it better be uncharged. So is arginine going to fit in there? Nope, that's not a good one. What about alanine? That's a pretty good one. So alanine could fit in there, in which case elastase is going to take this RAFNSME and it's going to cut right here. Okay. Is it going to cut anywhere else? Well, phenylalanine, that's a large hydrophobic residue. So it's hydrophobic, yes, but it's a large residue. So that's not a good one. And asparagine. Well, that's a polar residue. That's not a good candidate. S-serine, polar. M-methionine. Well, that's a large hydrophobic residue, so that's not a good candidate. And then E-glutamic acid. Oh, but wait. Is there a peptide bond here? No, there is not. There's no peptide bond there. So even if glutamic acid did fit here, which is glutamic acid going to fit, or is it going to be a small hydrophobic residue? No, it's a large polar residue, so it's not going to fit there. In addition to that, there's no peptide bond after that glutamic acid. So what elastase is going to do to this is make this and this peptide separate from one another. Excuse me. Now, our last serine protease is that chymotrypsin. We've already seen what it does. The only thing that differs about this peptide and the peptide that I'm using here is an arginine. Chymotrypsin, if it sees an R here, nothing changes because R is a positive polar residue. It's not going to cut, or that's not going to influence it. Okay, so with all of that said, one of the cool things about the class of enzymes known as serine proteases is here are a total of 3 different serine proteases. If we look at chymotrypsin in the middle there, chymotrypsin's got histidine, aspartate, serine, tryptophan, and glycine. Well, those amino acids, those are all going to contort themselves and fold into 3 dimensions to make those 3 residues present so that they can break a peptide bond. These are the hallmark residues: histidine, aspartate, and serine. Those 3 residues are kind of the keys when it comes to breaking a peptide bond and doing the chemistry that I talked about in the previous video. That serine 195 in chymotrypsin is our nucleophile. Serine 146 and serine carboxypeptidase 2 is going to act as the nucleophile. Then serine 125 in subtilisin. So all of these residues, these 3 big ones, are conserved across all of these different proteases. And let me go ahead and serine 195, serine 221, not 125. His 64, aspartate 32. So those are our conserved residues. So those are the keys that make up the active site. I always like to look at an image or a model like this because you have these amino acids in their primary structure and they're really far away from one another. But then whenever that protein folds into its 3D shape, aspartate 102 is right next to serine 195, which is right next to histidine 57. Now, I know that we already went through the serine proteases, so I'm going to kind of breeze through this. It's that catalytic triad. Aspartate 102 just kind of hangs out to help histidine 57 stay in the right place. Serine 195 is your nucleophile. Now, what's lacking from the model that I showed you previously is just this glycine residue, this Gly 193 right here. What that's going to do is that is going to work alongside the N-termini of serine 195. And that's going to help form this oxyanion hole. And that oxyanion hole is going to basically help properly position our peptide. Properly positioned peptide. That's a onomatopoeia or alliteration. Yeah. One of those two, I can't remember. 9th grade, this is what I remember. This doesn't change or really, this doesn't majorly change the residues that are involved in breaking the peptide bond. But this helps to coordinate that carbonyl oxygen, position it so that these peptide bonds can be broken. But in the video group that I showed previously, I went through each one of the different players. Your takeaway: serine proteases. Serine is your nucleophile. Histidine is an acid-base chemistry, and aspartate 102 is really kind of its wallpaper. It holds everything together. It's if you've ever seen the movie The Big Lebowski, it really brings the room together. Now, another depiction of the active site where that oxyanion hole is depicted here. The N-termini of glycine and serine 195, that's able to properly position our peptide so that that can be broken. They just kind of work in concert with the other amino acids within the active site. Now, other proteases are classified in part because of one of the key residues that are in their active site. Cysteine, aspartyl, metalloproteases. Well, metalloprotease, obviously it's not a metal amino acid, but instead it's an amino acid or amino acids that help coordinate and position a metal ion. That's going to enable something like water to be captured. We saw with carbonic anhydrase what happened, but here what we've got is, in this model, if this zinc ion coordinates this water molecule, that makes this base so much more capable of picking up this proton because this water has a lower pKa. When that water has a lower pKa, now all of a sudden this lone pair of electrons, or this bonded pair of electrons, can go and pick up this carbonyl carbon. That carbonyl carbon can push these electrons out to that oxygen and get us closer to breaking that peptide bond. Cysteine proteases are going to have a cysteine residue in the active site. Aspartyl proteases are going to have an aspartic acid in their active site, and chances are they're going to use another aspartic acid as well. Now, the last protease, or not protease, but the last type of covalent catalysis that I wanted to talk about was the mechanism for an enzyme known as lysozyme. Lysozyme hydrolyzes the peptidoglycan layer of bacterial cell walls by breaking the beta-1,4 glycosidic linkage between NAM and NAG. That's N-acetamiramate and N-acetylglucosamine. These are two sugars that make up, well, the peptidoglycan layer of bacterial cell walls. Now, what's cool about this to me is that this is an enzyme that is, sure, it's breaking this bond and it's using water to do it, but there's a lot more to it. And this is, to me, this is one of the first times that I saw this as a student, where I said, okay, let's say that we have a protein that's 765 amino acids, okay? Then what we can do is someone's going to narrow down that there's like, in the case of a serine protease, there's, let's call it 4 amino acids. So those 4 amino acids make up the active site. Now, those 4 amino acids that make up the active site are, yeah, they're important. No doubt about it. What about the 761 other amino acids? Like, where are they involved? What's the significance of one of those being mutated to any other amino acid? Like, what's the deal there? And it was this figure right here on the right that really helped me understand that. And I'm going to flip ahead because this is the mechanism that we're going to look at. And one thing I want to draw your attention to is there are only 2 amino acids that are highlighted. And we will talk about this entire mechanism by looking at these 2 amino acids, glutamic acid and aspartic acid. Glutamic acid 35, aspartate 52. Now, I like to mention those because then I can highlight them right here. And I'm going to use the highlight function to do that. Aspartate 52, glutamic acid 35. And oh, this figure is great because it even says where we are breaking our bond. Okay, so we're breaking the bond right there. But then if we look back, we've got, let's start at the top. We've got aspartate 101. Tryptophan 63, tryptophan 62, alanine 107, asparagine 59, valine, that's a hydrocarbon. How is valine going to be involved? Oh boy, valine 109, glutamine 57, again glutamine, asparagine, glutamic acid, phenylalanine, asparagine, arginine. Oh man, ugh. That's a lot of amino acids. When we look at the mechanism, we're really only going to be looking at 2 of them. What's going on there? And the answer is a lot, because each one of these amino acids is going to be doing something different. To aspartate 101, for example, that is going to form hydrogen bonds with this group and this group. Now, with that in mind, there's also a pattern here. NAG, NAM, NAG, NAM, NAG, and then this last one that's kind of obstructed is NAM. The way that I always think about this is we've got basically that peptidoglycan polysaccharide, that really long sugar, it has to start somewhere. So it has to have sugar number 1, sugar number 2, 3, 4, 5, and 6, and so on. And it might be 300 sugars long. But the very first one is going to be NAG. The next is NAM, NAG, NAM. It's just going to go, it's a pattern. NAGNAM, 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 over and over and over again. Lysozyme is going to kind of process it. I'm going to use my straw again. It's going to process it. It's going to grab onto this end. And it's going to look at the first sugar and say, that's NAG. Next sugar, that's NAM. Okay, I don't care about those 2. NAG, NAM. Aha! I've seen 2 NAGs. I've seen 2 NAMs. I'm going to cut now after the second NAM, because I'm going to cut after NAM. Basically, I'm going to say nag, NAM, cut. Nag, NAM, cut. Nag, NAM, cut. So essentially, all of these amino acids, like this aspartate 101, is helping whenever our active site is positioned properly, it's helping to support this end of the sugar. And then, excuse me, arginine 114 is helping to support this other end of our sugar. And whenever the time is right, which I really wish for dramatic effect, certainly I had a pair of scissors. Oh, but wait, I do. Just saw them. Here we go. So nag, NAM. That aspartate and glutamic acid are right here over this cut site. And how do you like that? Then this aspartate 101. It's going to process and it's going to bind whenever that is coordinated with those hydrogen bonds or making those hydrogen bonds. That indicates that my aspartate and glutamic acid are over the site where they're going to cut. So they're going to cut. Okay, so what I like about this is basically this is illustrating how everything else is foundational. Everything else, all these other amino acids are supporting the proper positioning of these bonds. I think that's so helpful and so powerful because it shows that, you know, yeah, you might have two amino acids that are key to catalysis, but you got 763 other amino acids that play a role. And they might not be directly involved in the catalysis or breaking that specific bond, but they're kind of, those active site residues are sort of standing on the shoulders of giants in some respects. Okay, so now what we have, let's get down to the brass tacks of these two amino acids. We have glutamic acid 35. We have aspartate 52. If you look at this very first step where our sugar is bound, this bond right here, these electrons are going to pick up that proton. This glutamic acid was protonated. It was acting as an acid. And from here to here, it goes from protonated to deprotonated. So uncharged to charged to negatively charged. That's going to leave behind a substrate that has a positive charge. It's an oxonium ion. This is absolutely general acid catalysis. Then what we've got is aspartate 52. That's a negatively charged residue. Ooh, aspartate 52. Negatively charged. Man, if something's negatively charged, and actually, let's go back and let's revise this for a second. Hydroxyl group. If you see this, I mean, that's a hydroxyl group right there. That is. So I think we should modify this or amend this and throw on our aspartic and glutamic acids. D and E can have that hydroxyl group and have it form that nucleophile, which is exactly what we have right here. That aspartate 52 forms a covalent bond with our substrate. So this was originally deprotonated. It remains deprotonated until we form this covalent intermediate. Now, this is where, when we talk about our major types of catalysis, acid base, metal ion, let me hear it. Saw Metallica a few years ago. Metal ion catalysis. Well, when we have that, sorry, and then our last is covalent. Our last major one is covalent catalysis. We've already had a general acid. Now we have a covalent intermediate. So how would we classify this as acid-base catalysis? No, we'd classify this as covalent catalysis because as soon as that covalent bond is formed between an amino acid and our substrate, it becomes covalent catalysis. Water is then going to come in because what water is going to help us do is water is awesome because it can act as an acid, it can act as a base. It's going to act as an acid for glutamic acid 35 or glutamate 35. So it's going to protonate that glutamate to make it glutamic acid. So here's our proton back. Then the remaining hydroxide group is going to go on to that carbon. That's going to cause our aspartate 52 to break that bond, to break that covalent bond and release our sugar. So water is acting as a base in order to do that. So this is an example of covalent catalysis, another example of covalent catalysis. An example of covalent catalysis where we have acid-base catalysis, but it being covalent catalysis trumps it all. Okay. Now, I hope this is helpful. I would definitely know the different amino acids that can participate in different types of catalysis. The sorts of questions that I like to provide you with are, what is this amino acid acting at? And then I also like acting as. And so I will draw it. If an amino acid, if let's say an aspartic acid, for instance, is protonated, chances are it's going to act as an acid and donate a proton. So think about the protonation state of an amino acid, and is it going to act as an acid or a base? That's the sort of question I really enjoy asking. I hope this is helpful, and I hope you have a good one.